Hello nerds, today we are going to study about bacterial growth curve. Growth means that they are going to increase their number of cells. If the number of cells are increasing, it means the bacteria is growing. The bacteria, they increase their number of cells by dividing the cells into two new cells. There are four distinct phases of bacterial growth curve. The first one is lag phase. To study the lag phase, let's take the example. You have a test tube, and in the test tube you have added this yellow medium. Now this yellow medium is actually the growth medium. It means that this medium has all the necessary nutrients for the bacteria to use and divide into cells or into new cells. So in this medium you have added some cells here. For example, we have added 100 bacterial cells. At this time, this phase, when you have added the bacterial cells in the growth medium, it will be known as lag phase. This is a phase in which no increase in number of bacterial living cells will take place. Why? Because at this time the bacteria is actually adjusting to this environment, and it is not dividing but understanding where it is present. So because of this reason, no bacterial cell division will take place, and if you have added 100 cells, so during the lag phase there will only be 100 cells. So this is the lag phase. After the lag phase, as the time passes, we will have log phase. In the log phase, what will happen? The exponential increase in the number of living bacterial cells will take place. It means now the bacteria has adjusted to its environment and it is started to divide, and as it divides, its number of cells increases with the passage of time. So as the time increases, the number of cells increases. So this phase is known as log phase. You can clearly see here the medium was very clear, but as the number of cells starts to increase, it has become very dense or very cloudy. So this is going to indicate that the bacteria, they are dividing or they are present in the log phase. After the log phase, we will have the stationary phase. The stationary phase is actually the plateau or a straight line. Why? Because it is going to represent the number of living bacterial cells. Now at this stage, the rate of cell division and the rate of death is equal. For example, if 15 cells they are in dividing, 50 cells they are dying. So in this case we have the straight line. Although we have more cells here, but the new cells and their death is equal, and because of this we have this straight line, or we can say that now the phase is static. It is not increasing and it is not decreasing. So here you can see very cloudy in comparison to this one. This tube is more cloudy. Why? Because now this is in the stationary phase. Then comes the last phase, which is known as death or decline phase. This is the phase where the number of dying cell has increased. It means now the cell division is decreased, and now the cells which were present in this area or at this time, they are going to die, and because of their death we are having less number of bacterial cells in the medium. So this is the phase of death or decline. So we can say that exponential decrease in the number of living cell is known as death or decline phase. So in summary, we have four phases of bacterial curve cells. One is lag phase, when no increase in number of living cells takes place. Then comes the log phase, when it going to increase. After that we have the stationary phase, when the number of dividing cells and number of dying cells are same. And then comes the death or decline phase, when the number of dying cells they have increased, and because of this the number of living cells is going to be decreased. That was all about the bacterial growth curve.